Hello, everybody. I'm really delighted today to have with us Pamela Ohashi. So I'm going to introduce Pamela. Hello, Pamela. Hi, how are
Hello.
you?
Good, thank you. Nice to be
So,
here. How are you?
thank you, Pamela. So, Dr. Ohashi received her PhD from the University of Toronto with Dr. Tak Mak, and then her postdoc training. She did her postdoc training at the University of Zurich with the Nobel laureate, Dr. Zen Kanager and Dr. Hans Hangartner. She is a senior scientist currently. She is a senior scientist and director of the Tumor Immunotherapy program at the Princess Margaret Cancer Center and a professor in the Department of Immunology at the University of Toronto. Her interests include in understanding CD8 positive T cell biology and also the mechanisms that regulate anti-tumor immunity. So we're really happy to have you today with us, Pamela. So she's gonna talk to us today about immunoregulation and the tumor microenvironment. I just wanna tell you guys that you can ask questions in the Q&A tab, and you can also evoke questions of other attendees. I will ask actually those questions at the end of the webinar verbally to Pamela. I'd also inform you, I would like to inform you that the webinar will be recorded and that you can view it again later on demand. Donc, final point is I would like to thank Thermo Fisher Scientific for supporting this today webinar. So thank you very much, Pamela Ohashi, for being with us today, and I give you the floor. Thank you.
Thank you very much, Abdallah. Okay, so there we are, back at the beginning of the slides. Let's see. Oh, it looks like, okay, good. So, hello everyone. Not sure what time zone everyone's in, but nice to have you all join us today. I'd like to give a kind of a different talk today. I think what's been exciting for me is really to see how cancer immunotherapy has really come so far in terms of cancer therapy, and it's been really fun to watch how exciting this field has grown in terms of many different strategies. I'm going to focus today on PD-1 blockade, and what we were interested in the lab in doing is looking at different cancers and trying to understand why some respond and some don't respond. And so I wanted to share some data with you today, just with the message that there's a lot for us to learn in cancer biology and immunotherapy, and the different ways that we can really move this field forward and propose rational combination therapies. So I'll start by really just giving a very basic overview of the signaling pathways and the molecules just that I'm talking about today. So on one side, we have the T cell, and of course, that's really the critical engagement there, or the specificity of the T cell interaction, is dictated through the T cell receptor, which recognizes a peptide and MHC complex on the side of the antigen presenting cell or the target cell. And if we're thinking in terms of human biology, of course, this is an HLA restricted peptide interaction. This conversation between the T cells and antigen presenting cells includes many molecules, but here I'm just focusing on PD-1, which is known to be a negative signal that goes to the T cell. PD-1 engages two ligands, PD-L1 and PD-L2, and these ligands can be expressed on many different cell types, in particular for PD-L1. So of course, it's expressed on the antigen presenting cell, as I've shown here, but PD-L1 can be expressed on tumor cells, endothelial cells, cardiac myocytes, and other cells. Just by way of introduction too, I think some of the key things that we think about when we think about PD-1 is we know that it's a marker of both activated T cells as well as overstimulated or exhausted T cells. And so it plays a role in the biology at least in 2 time points when it's trying to regulate the T cell interactions and the signaling going to the T cell. PD-1 is also expressed by many other cell types, and I think in the field of cancer biology, we really think primarily about the T cell and the role of PD-1 on T cells, but I would just like to raise the point that PD-1 is also expressed by many other cells, including NK cells, gamma delta T cells, B cells, and other cells. So to have a complete picture of what's going on when we administer PD-1 to patients, I think we should consider what's going on with these other cell populations as well. It's also, there's some nice studies that have been reported as well, implicating PD-L1 in induced Treg populations, and that this is important for maintaining the transcription factor FoxP3 expression. So this is just a very quick overview of PD-1 and PD-L1 interactions that are some of the things we should consider when we're thinking about immunotherapy. So here's a slide just highlighting some of the, I think, important reports on the response of various cancers to PD-1 blockade. And these are really over the past 10 years already, that there have been all these reports that really have revolutionized the field of cancer immunotherapy. And I think many of these were very surprising, that bladder cancer, non-small cell lung cancer were really responding well to PD-1 blockade as a monotherapy. But you can see really that there's quite a range of response rates, from 13% all the way up to 87% in different types of cancers. So going forward, I think what is important in the field and what people are really thinking about is, why is it that these cancers are the ones that respond? How can we make them more durable? What are some of the things that we can combine with these, with PD-1 blockade to really improve cancer immunotherapy going forward? And so some of the things we were thinking about in the lab was really, can we identify factors or mechanisms that would explain why some tumors don't respond well to PD-1 blockade? And so this is some of the data I'd like to share today. And one of our approaches was to compare ovarian cancer and melanoma, because ovarian cancer is known not to respond that well to PD-1 blockade, whereas melanoma does. And so we thought this would be a nice way to look both from a positive and a negative responding patient's perspective, or tumor perspective. Okay, so some of the attractive things about ovarian cancer is that this is a report by George Koukos's group quite some time ago now, but they showed that overall survival in ovarian cancer is linked, and this is epithelial ovarian cancer, is linked to the number of T cells that they can see infiltrating the cells, the tumor. And so if there was a lot of intratumoral T cells, the patients had a better prognosis in terms of survival, and compared to when there was no or few intratumoral T cells. And so many of these T cells have been shown to be CD8 T cells, and Brad Nelson's group has really taken this a bit further and shown that both B cells and CD8 positive T cells are a good prognostic factor for ovarian cancer, which then we thought, well, okay, so if we know CD8 cells are responsible for protecting patients or improving survival, then maybe it's just another little push that we need to figure out to get these patients to respond much better to PD-1 blockade. So we felt that they're just hanging on the edge, and it would be really nice if we could figure out how to push them over and get responses to PD-1 blockade. The other thing that was really attractive for us for looking at ovarian cancer was the patients, unfortunately, since they present so late, they also tend to relapse also quite quickly. And so by looking at some of the factors, we were able to, or we thought we could get a better handle on the relapse rates in these patients as well. So these are studies done by Aras Toker, and one of the first questions we wanted to ask was when or what cells are actually expressing PD-1 in the ovarian tumor microenvironment. Okay, so what Aras did was we used very many different markers to stain cells. These are cells that were viably frozen from ovarian cancer patients, so no expansion, no anything, just viably frozen, and then we thaw them and look at them. And he used many markers, but I want to show this one here where we're looking at PD-1 expression on the y-axis and FoxP3 on the x-axis. So we had the impression that it was the regulatory cells that were expressing intermediate levels of FoxP3, and that it very well defined these regulatory cells. And so this is gated on, again, CD3 positive cells and CD3, CD4 positive cells. So you can see that there's a nice population that shifts to the right that are PD-1 intermediate and FoxP3 high. And if we stain many, many, many different ovarian cancer samples, we really see this as a reproducible phenotype of these cells. And so if you just look on the right, we compared the expression of PD-1 from the regulatory T cells in the tumor versus PD-1 expression on regulatory T cells from the peripheral blood. And on the y-axis here, we're looking at the mean fluorescence intensity of PD-1. And you can see that in the tumor, all the regulatory T cells are expressing some level of PD-1, whereas when we look in the peripheral blood, we don't really detect expression of PD-1. So we were surprised that there was this change depending on the site of the tumor, the site of the regulatory T cells. So we interpreted that to mean that there is some kind of activation or something is going on inside the tumor that's leading to either the activation of these regulatory cells or recruiting these regulatory cells. So then our next question was, how does this look in melanoma, actually? And so here we're looking at 3 different patients going across, and then we're looking at 3 different populations of regulatory cells. Regulatory cells from the peripheral blood on the left, regulatory T cells from the ovarian cancer samples, and then also from melanoma on the far right. And so again, you can see that the regulatory T cells in the blood don't express PD-1, which is the x-axis. Ovarian cancer samples, they do express PD-1, and it's quite at a high level, easily detected. But what was interesting we found was that the melanoma regulatory T cells really do not express PD-1, and it's very, very low levels. And this was very, very reproducible, again, between different tumors, which we thought was quite unexpected. And so here we're quantifying, each one of these dots is a different sample from a different patient, and we're looking really at the differences now between ovarian cancer and melanoma. So again, on the y-axis is the mean fluorescence intensity, and we're looking at, actually, yes, PD-1 expression on the left set of data, and so showing that this is, again, very reproducible, that the regulatory cells from ovarian cancer really express quite a higher level of PD-1 compared to melanoma. And on the right figure, we're looking at FoxP3 expression, showing that the ovarian cancer Tregs are really much more activated than the regulatory T cells from melanoma, as judged by the expression of FoxP3. So this was really surprising to us from a couple of perspectives, particularly that, one, we could really find a identifiable phenotype within a certain cancer type, that is ovarian cancer versus melanoma, and that they were really quite characteristic of being from either of these cancers. And so here I'm summarizing a lot of the data because this has been published a couple of years ago, but this was really some of basically the bottom line, and that is regulatory T cells from the peripheral blood really don't express a lot of activation markers, and they're not very immunosuppressive. We found that the regulatory T cells from high-grade serous cancer expressed a lot of these activation markers, and we looked at many more than one I've shown here, but here we're showing PD-1, 4-1BB, and ICOS. And then melanoma regulatory T cells really are not that activated. They're more slightly higher than in the peripheral blood, but again, very different from the regulatory T cells coming from high-grade serous cancer. And when we look at the function, again, the regulatory T cells were the most suppressive when they were coming from high-grade serous cancer. So what does this mean, and what are our thoughts about these first experiments? One of the things that it predicts was that maybe the Tregs are more activated from high-grade versus, well, that was our interpretation, that they're more activated in high-grade serous cancer versus melanoma, that PD-1 expression can indicate the activity, and that maybe we should be considering some other things when we're looking at patients treated with PD-1 blockade. So about a year after we published this, there was a study by Nishikawa's group in PNAS, and they were asking the question whether these regulatory cells expressing PD-1 really play into the biology, and in particular of hyperprogression. So there's this state, a hyperprogressive state that has been reported in in some situations where when patients receive PD-1 blockade, that they actually really get worse quite quickly. And this is known as hyperprogression. And this study showed that when they looked at biopsies from these hyperprogressive patients, that they actually saw more regulatory T cells in the biopsies from these gastric cancer patients. So this is consistent with the idea that these Tregs are actually getting more proliferative and more inhibitory and leading to this hyperprogressive state. What was also intriguing to speculate was that we wanted to ask the question actually, or we speculated that if you block both Tregs and give Nivo in high-grade serous patients, would that actually improve their response rates to checkpoint inhibitors? And so one of the things that has also been reported is that IPI can actually result in the death of regulatory T cells, which is mediated by non-classical monocytes. And this was studies by Daniel Spizer's group. And so if you put this all together and do a combination trial, it might be that if you combine IPI and Nivo together, the high-grade serous cancer patients would do better. And in fact, that was what was recently reported in JCO by Dmitry Zamarin's group. They showed that single-arm patients, so patients receiving Nivo only, responded at a rate of 6 out of 49 patients, whereas if patients were receiving both IPI and Nivo, they saw a response rate of 16 out of 51, suggesting that those patients were doing, of course, a little bit better in terms of the overall response rates. So I think these studies really show that if you think outside the box and consider other things related to PD-1, that we could think that these PD-1 positive regulatory cells are linked with hyperprogression, and also you can improve response rates if you add in other combination trials such as IPI-Nivo. I think these are all speculations and that they have to be further studied, but it's consistent with those findings or interpretations. So one of the family members that is really prominent in terms of targeting for checkpoint blockade is the B7 family member. And so I've outlined these different members in this slide here. And so on the top, we have the antigen presenting cell again, the bottom, the T cell. And aside from the key B7-1 and B7-2 molecules, which are CD80, 80 and 86. We've also got PD-L1, which we just talked about, but there's other B7 family members, namely B7H3 and H4, that are attractive targets really to think about combination immunotherapies. And so I'd like to talk about a little bit about those in the next couple slides. But in terms of ligands and receptors on the side of the T cell, I think some have been reported, but by and large, it's really not known what B7H3 and B7H4 are binding to on the side of the T cell. I'm going to skip this slide and just make a very, very brief introduction about both of these molecules, B7H3 and B7H4. There's many names that these molecules have been called over the years, and so I just wanted to show them in case somebody thinks of them in terms of other names. But what's really known, there's a lot of work that has been done on these molecules, but very briefly, B7H3 has been involved in both enhancing and inhibiting T cell function. B7H4 is more an inhibitory molecule, and that is in studies where they blocked interactions with B7H4, they could show that there's increased T cell function, and that is read out as either proliferation, cytokine production, or cytotoxic function. Both of these molecules are known to be expressed by the tumors and associated with poor prognosis. And so it's really, I think, one of the attractive targets for immunomodulation for immunotherapies. And so we thought it would be helpful or insightful really to have a look at what cell types really express these molecules. And these are studies done by Heather McGregor, and we were looking at B7H3, B7H4, and PD-L1 in the context of ovarian tumors, and asking the question, what different cell types express these molecules? And again, very straightforward experiments. These are fresh specimens from debulking surgeries. viably frozen, and then we thaw them up and look in different ways. And she did flow cytometry for her analysis and immunohistochemistry. And what I'm not going to talk about is the TCGA data, but we've done that, and can check our papers if anybody's interested in that aspect. But again, what I want to focus on today is really the unexpected findings that we have seen from evaluating ovarian cancers. And so just focusing in on that data for this talk. So here are 3 examples of the types of patterns of expression of B7H3 that Heather has seen. And so we've got the immunohistochemistry on the top, but on the bottom you see that on the y-axis is forward scatter, and then we're looking at B7H3 across the x-axis. And so the red indicates the B7H3 positive cells over background, and you can see that B7H3 actually identifies 2 different populations in some samples, and only one on the far right. But when we saw this, we thought, well, let's try and identify what different types of cells are expressing B7H3. And so when she looked at epithelial markers on the left, E-cadherin or EpCAM, you can see really that B7H3 is not expressed on the epithelial cells very highly. But when you look on the stromal cell markers, and here we have FAP, protoplanin, PDGF receptor beta, and CD10. That's the cells that are really expressing high levels of B7H3. And for us, this was really unexpected because the literature was saying it's all on the tumor cells. And so let's go to the next slide. Here we're quantitating the amount of B7H3 that's being expressed by mean fluorescence intensity, and just showing that over all the different samples of tumors that we were looking at, B7H3 is really low on the epithelial cells, but very high on the stromal cells. So this then led us to appreciate the fact that B7H3 actually identifies stromal cells. So the B7H3 high cells are the stromal cells. And we asked the question, what happens if we stain with both B7H3 and B7H4 at the same time? And then I thought we got a kind of an interesting profile that looks like what we have on the left. And that is the tumor cells are showing in the red that they're B7H4 positive, and clearly segregating out the stromal cells, which are B7H3 high. And when we look at the single parameter analysis on the right, the tumor cells are really expressing B7H4 at high levels, and stromal cells are not expressing B7H4. So this conveniently is a very nice marker to look at tumor cells in the ovarian cancer tumor microenvironment. So we're quite convinced that if we just gate on B7H4 cells, that we're really looking at the tumor cells. So this allowed us to ask the question, what cells are expressing PD-L1? And so here we're looking at 2-parameter analysis for B7H4 on the far left, so on the y-axis, and PD-L1 on the x-axis. And you can pretty much see that in ovarian cancer, there are really no tumor cells expressing PD-L1. And I think for those of you who have followed PD-L1 expression over the last 10 years, first of all, it was thought to be a marker of patients that would respond well to PD-1 blockade. They've also shown that then PD-L1 can be expressed on macrophages, and that was a good indicator of response rates. But here, I was surprised to see really that no tumor cells are expressing, essentially no tumor cells are expressing PD-L1. And turns out, if we use our different markers, we can show that PD-L1 is being highly expressed on the stromal cells, which is on the middle panel. And then again, on the right, the tumor cells are expressing the B7H4 at high levels. And again, so this was very unexpected because not a lot of people talk about what is the implications of PD-L1 being expressed on stromal cells. So just in summary, the points that I wanted to highlight from these studies is that for us, unexpectedly, B7H3 is highly expressed on the stroma. We also find it on tumor cells, on antigen-presenting cells. But if you read the literature again, most of the times you read about high B7H3 on tumor cells is a bad prognostic indicator, and nobody really talks about the stroma. Although it has been reported in other cancers as well, I should also mention. B7H4 is highly expressed on tumor cells, but by looking at the combination of B7H3 and H4, we were able to really evaluate what cells are expressing PD-L1. And again, it's predominantly not on the tumor cells, but on the stroma. So then this really begs the question, well, a couple points comes from this. Well, so if people are thinking of targeting B7H3, actually, at least in ovarian cancer, the prominent population is the stroma. So that whether or not stroma is going to be important for modulating, and it could be, because there's lots of literature saying that stroma is bad. So it just gives another avenue and another mechanism how anti-B7H3s might be working when the studies are done down the road. I think also we have to consider that PD-L1 being expressed on the stroma. I think it begs the question, does this identify excuse me, patients that do not respond that well to PD-1 blockade? I don't know. I think it's something that can be looked at down the road, looking at other cancer types that don't respond well. Maybe this is a common feature of some of them. Nonetheless, it's suggesting really that the biology around the tumors is very different, I think, in ovarian cancer. I'm just checking on the timing here. So yeah, I'd like to spend the last bit of my talk talking about innate lymphoid cells. And I just give a very brief introduction on these cells as well. So on the left, innate lymphoid cells have been grouped into three groups based on the cytokine expression. And so we have group 1, which includes the classical NK cell, I think the prototype cell that has been studied for years. And we know that this cell is cytolytic and expresses CD56. But because it expresses interferon gamma, it's been lumped into the group 1 category. Group 2 expresses cytokines 4, 5, 9, and 13, and group 3 expresses the cytokines shown there, lymphotoxin TNF, IL-22, and IL-17. So we did some studies in ovarian cancer, and this is summarized on the right, looking at tumor-infiltrating cells and their ability to grow and how fast they grow in culture. And it turns out that we found a CD56 positive cell that we called an ILC-Reg that was responsible for inhibiting the growth and proliferation of both the CD4 and CD8 cells. And we found that this was due to an interaction through NKp46. So on the right, this is a summary of the phenotype of the cells that we found. They expressed CD56 and could secrete IL-22 and IL-9. So if you look back to the left, the categories of the innate lymphoid cells, the cells that we were finding in the tumor microenvironment actually took one feature from each of the groups. That is CD56 from group 1, IL-9 from group 2, and IL-22 from group 3. So we're not quite sure what to call these cells, but nonetheless, they really had a regulatory type capacity. And so there's really quite some literature on ILCs, of course, and their impact on tumor immunobiology. But here I'm highlighting just 3 different papers that came out about the same time in 2017. So showing that these ILC-Regs have an impact on the tumor microenvironment, which is what we showed on the left. But other groups have also shown, Wang's group Kwong as well, that these cells are really important in mediating inflammation. And so there's an ILC-Reg looking at intestinal inflammation that produces IL-10 and regulates ILC-1s and 3s. And this regulates then intestinal inflammation. And in terms of neuroinflammation, there's a population of cells that act very similar to ours through NKp46 to regulate CD4 cells and again also regulate inflammation in that case. So we thought it would be important to really see if these cells are found in other cancers and what are some of the properties, and can we find defining features of this particular population of cells. So Catherine Warner took on this project and we looked at ILCs in breast cancer. So here were what some of the first studies that we did was she was asking similar questions as we did in ovarian cancer. We're looking to see how fast different TIL cultures grow. So again, surgical resections, put them in culture for here 4 weeks, 4 to 5 weeks. That's the x-axis, and we're just counting the number of cells over time. And so generally speaking, what we call a fast-growing cultures are the open circles, and the slow-growing cultures are the darker blue circles. And when we ask the question in the slow-growing cultures, what's the percentage of CD56 positive, CD3 negative cells? So these are the cells that we were calling ILC-regs in ovarian cancer. We see the same correlation. So the amount of CD56 positive cells in the slow-growing cultures are really much higher than the proportion of the same cells in the fast-growing cultures. So consistent with the correlation between these cells and slow growth of cultures. And we found something else that was different about breast cancer, so I'm just highlighting it here. And we're looking at the number of CD4 positive cells in fast and slow growing cultures, which is on the left, and the number of CD8 positive cells in the fast and slow growing cultures, which is on the right. And you can see really in the fast growing cultures on the left, there's a predominant population of CD4 cells that I think is really quite striking in the fast growing cultures. And in the slow growing cultures, you can see that it really skews, which makes sense, to really less CD8 cells. And so for some reason in breast cancer, we really see that there's a predominant CD4 population in those cultures that are able to expand very well. You see the trend in the slow growing cultures as well, but it's statistically significant on the side of the CD8s. Actually both sides, but it's visible, it's very visibly striking, I think. So I want to come back to this, that it's a unique property really of the breast cancer cells. So asking the question whether these CD56 positive cells are actually inhibitory, Catherine did a few more experiments to show that they're actually a kind of a bad thing to have in your cultures. So on the left, we're looking at fold expansion of the TIL cultures, just with the CD56 cells sorted out. So we sort them and we add them back and ask the question whether they're inhibitory in terms of proliferation. So if there's no CD56 positive cells in the cultures, you could see that the culture grows better. And if you do a more traditional regulatory type assay that's on the right, you can see that they do have a regulatory function. And so just to describe this assay, we take the TILs, load them up with a dye, and then stimulate them with anti-CD3 and 28, and then add back the slow-growing cells, the CD56 positive cells or not, and ask the question whether the presence of those cells inhibit the proliferation of the TILs. And so on the graph on the right, we're showing the percent suppression of the CD8 positive cells. And so in the absence of the TILs, they do proliferate well, but when we add back the CD56 positive cells with the TILs, then you can see that there is an inhibitory function in terms of them blocking the suppression of the TILs. So this is all consistent with the idea that these CD56 cells are inhibitory and block proliferation of these cells from two different types of assays. We characterize the different cytokines that are being expressed in these cultures. So in this experiment, what Catherine did was, these are just the fast and slow growing cultures, and then we take the supername and ask the question, what kind of cytokines can we detect? And what was quite different we did this both in the ovarian cancer cultures, but in the breast cancer cultures. And then you can see really here, we're seeing a lot of IL-5 being produced. We don't see this cytokine coming up in ovarian cancer. In terms of IL-9 and IL-22, you can see that there is some being detected in these cultures. But this profile, again, is very different from the ovarian cancer cultures because although there was more IL-9 and 22 in ovarian cancer, called TIL cultures, there was really quite a bit of expression of these cytokines. So the fast-growing cultures on the left would really have a lot of production of these cytokines, both 9 and 22. What we saw that was also quite different was when we looked at Granzyme B, there was a lot more Granzyme B that we could detect in the slow-growing cultures, shown on the right. Okay, so we had previously reported that IL-9 and 22 were being produced by these ILC cells. And what Catherine went on to show was that this is actually dependent on the T cells in the culture. So you basically have to co-culture TILs together with the CD56 cells in order for the CD56 positive cells to make these cytokines. And that's shown in this figure. The top is the regulatory CD56 cells only, and the bottom is the regulatory cells together with the TILs. And you can see that only in the bottom do we detect IL-9, IL-22, and IL-17A really serves as a control for this particular experiment. So this is something I think that really gives us a little bit more understanding about how these cytokines are being produced. What we also looked at was different markers being expressed by these regulatory type ILC cells, and she really looked at, I don't know, 20, 30 different markers, and I highlighted what we think are the most interesting ones in the slide. And so on the left, we're looking at CD7 expression on the regulatory cells from the slow-growing cultures, and comparing to the CD56 positive cells that we can detect in the fast-growing cultures. And so, but we can show only the ones that are inhibitory, or the ones in blue, or the ones coming from the slow-growing cultures. So that's what we're comparing here, CD56 positive cells from slow and fast-growing cultures. And in the ones that are more regulatory, that can inhibit proliferation, and again in the blue, we can see that they're expressing higher levels of CD7. This is really what we saw in ovarian cancer, that there was a shift, and that the higher levels of CD7 is really associated with the regulatory type CD56 positive cells. However, this is not strong enough, we feel like, because it's really just a minor shift in the intensity of CD7 expression, or the proportion as well. So then when we look at 2b4, we also see a shift to the cells, the CD56 positive cells that are regulatory express higher levels of 2b4. That's the middle panel. And on the far right, we're looking at TIM3 positive cells, again from the fast-growing cultures or the slow-growing cultures, and the slow-growing cultures with the regulatory CD56 positive cells have a higher level of TIM3. So we think this is a pretty good marker to distinguish the fast and slow-growing cultures, or the CD56 cells coming from the fast and slow-growing cultures. Okay, so in summary, we've looked at ILC regs now in both breast cancer and ovarian cancer, and both limit TIL expansion. In the slow-growing cultures, we have altered cytokine production, so suggesting that maybe there's something from the tumor microenvironment that's being detected when we grow these TILs, and they're already kind of skewed to a different kind of culture. And so that we're seeing more IL-5, a little bit of IL-9, and a little bit of IL-22. And this is different from ovarian cancer, where we see a lot of IL-9, 22, and not a lot of IL-5. The production of IL-9 and IL-22 is T-cell dependent. We showed that just by in vitro analysis, of course. And then in terms of lineage-defining markers, these ILCs express CD56, CD7 at a high level, 2b4 at a high level, and TIM3 at high levels. Overall, these are similar to ILC regs found in ovarian cancer TIL cultures, but there are different properties. So I want to come back to this idea that we saw a skewing of CD4 in the breast cancer cultures. And so this is really saying that the microenvironment is somehow different, that we can capture these differences by growing these TILs. And for some reason in breast cancer, they are more skewed to the CD4 lineage. I don't know biologically what that means at the moment, but I just want to emphasize that this is a different type of tumor with a different type of immune infiltrate, but nonetheless, we can really isolate these ILC regs from these two different types of microenvironments that still look like they have the same biological capacity. Okay, and the other thing I would like to point out is that in ovarian cancer, I didn't show the data, but the reason why we're intrigued about these cells is that when we look at the time to recurrence of these patients, so these patients were not treated with any type of immunotherapy. We simply just cultured their TILs, and the patients that have these ILC regs, and therefore the slower growing TILs actually recurred much quicker than the patients that did not have these ILC regs and had fast growing cultures. So, and the difference was actually 2 years in terms of the average time to recurrence. So we feel that there's a correlative analysis using these regulatory cells that really might have an important biological implication when we look at these patients. So that's why we're very excited still about following them up and understanding what the implications of having these cells are in the tumor microenvironment, but also in the context of immunotherapy. So to come back to the question that we asked in the beginning, why don't some cancers respond well to PD-1 blockade? Well, we've come up with a few things. Don't know really if they're key biological indicators, but I think it's interesting to consider that regulatory T cells are more activated in ovarian cancer and melanoma, and whether or not this actually plays a role in creating a barrier for responses to checkpoint blockade. I think that's one of the things that I think are in the forefront, at least of our kinds of thinking. So we're looking in different types of cancers to see what is the phenotype of regulatory T cells in different cancers, and then does that in the end correlate with other cancers that don't respond well to immunotherapy. We've also come up with these cells that we're calling ILC regs. It looks like they definitely interfere with things when we look in vitro, and they may interfere with the tumor immunity, I think, in the patients. And that's something we would like to further investigate. Some unexpected findings that I'd like to highlight as well is that there's a similar phenotype of cell within each cancer type. And I think going into this, biologically, we thought every cancer is going to have so much heterogeneity that we're not going to be able to make common conclusions based on cancer type. And I completely changed my mind on that. Like we clearly see that, for example, in breast cancer, there are bias to CD4 cells, and this is actually overgrowing many different TIL cultures. And this is lots of work done by Lin Nguyen. It's very clear that this is a phenotype really associated with breast cancer cells. And so that's one example, but then when we look at the Tregs from ovarian cancer, we also see that this is a very conserved phenotype. So I really do think that we can understand a lot about the biology of human cancers by really looking at these different tumor types and comparing them between different, for example, good responders and not so very good responders for immunotherapy. Some other unexpected findings that I have highlighted along the way is that we're not expecting that B7-H3 is most highly expressed on the tumor stroma. And I think this has implications if we're thinking about immunotherapy using targeted reagents for B7-H3. And the other thing that was unexpected really was the level of PD-L1 expression on the stroma. And particularly, we've only really looked in ovarian cancer. So whether or not this really impacts the response or changes the biology or is an indicator of the biology, I think is really something that still needs to be explored as well. Okay, so I think I would just like to acknowledge all of our collaborators that really were crucial in helping putting together this story. All the people in the middle there were our surgeons and oncologists and pathologists that have been critical for helping us collect the tissues. We're grateful to the patients and their families for allowing us to ask research type questions with these tissues. And Louis Lanier and his group were really critical in helping us look at these ILCs or NK cells, the big debate of whether or not they're NK or ILC cells. Anyway, we found them very interesting, and I'm happy to share these findings with you with help from Louis. Happy to take questions.
Can you hear me, Pamela?
Yes, I can.
Thank you very much for this great presentation. Really rich, very rich. We learned a lot today. Thank you very much.
Thank you.
So I have a lot of questions. I don't know how I'm going to handle all this, but I'm going to start right away. So the first question, Pamela, is related to the expression of PD-1 in innate lymphoid cells. So the question says, does ligation of PD-1 on ILC induce innate lymphoid cells, induce regulatory innate lymphoid cells?
Yes, so great question. We've looked at the levels of PD-1 on these cells, and it's actually relatively low. It's there, but it's relatively low. And so we haven't done a lot of those experiments yet. So I can't, I think it's a great question, but we have no insights at the moment.
Okay, thank you. A second question is related to the expression of PD-1, but this time on NK cells. So basically, they don't regularly express. This is what the person is saying. Basically, they don't regularly express PD-1, but can upregulate other immune checkpoint molecules, such as TIM3. So the question is PD-1 restricted to specific NK subsets or cancer entities? So this is the question, Pamela.
Also a very good question. Sorry, I have no insights on that one as well. But yes, we know that they are expressing these molecules, and I think that's the next. Those are the next steps. We really should understand the importance of these molecules going down the road. But other people have shown that TIM3, for example, on NK cells is an inhibitory factor and mechanism. So there's other examples of that out in the literature, but in terms of what we know right now is still early days.
Yeah, so another question is related to B7H3. So she was first described as a co-stimulatory molecule, but then claimed as co-inhibitory.
Yes.
So its role seemed to be controversial.
Yes.
Have you performed any functional assays? So the
No.
question is related to the functional assays.
Yes, I think so. Very good on that one too. So I appreciate the controversy in the literature, and I think so. The answer is no. We have not done any functional assays in the context of stromal cells in the tumor microenvironment. We have done other assays in the mouse, and I can say this is much more complicated than I really want to get into. But the bottom line is, in my hypothesis actually, is that I think B7H3 as in, so for example, with CD28, there's an inhibitory molecule CTLA4 and a co-stimulatory molecule CD28, and they're binding to the B7H is on the other side, CD8086. So I'm just thinking that B7H3 also will have a positive and negative molecule on the T cell side. We haven't figured out what those are yet. And so that's why I think that there may be still some controversy there is controversy in the literature about that. Having said that, we have looked at also the role of B7H3 in mouse models, and also in the context of NK cells or these regulatory cell types. And it's something that I didn't really have time to present here, and it's also a story in progress. But we know that B7H3 is actually regulating the activity of the ILC regs or the NK cells. Okay, so there is a really complicated story in the back going on about B7H3. So not only do we have to think about them on the stroma, but they're also going to have a role on the ILCs as well. So stay tuned.
Okay,
That's
Pamela,
we're working
thank
on
you. So actually another related question always to B7H3. What are typical ligands for B7H3 and H4?
Oh, there's
typical ligands.
Sorry, so receptors probably.
Yeah. Well, he said
So,
typical ligands, but yes.
Yeah. So B7H3, to my knowledge, nothing has been identified. B7H4 in the literature, there's maybe been 3 or 4 different papers that have been, and now off the top of my head, I don't remember what the other side is. More recently, they've shown one in JI, and this is Miller's group. Still, these receptors come and go, and no one has proven beyond a shadow of a doubt that these are the real receptors for the B7 3s and 4s. So, 4 in this case. So, I don't know yet. It's controversial, and we'd have to go back in the literature and look at. There was 2 or 3 that have been reported over the last 15 years, but by and large, most people say it's still unknown. So that's why I presented it that way.
Okay, another question is related to this very interesting difference that you've seen in terms of B7H3, differential expression in the stromal versus tumor cells. And the question says, how reproducible was the stromal versus tumor expression difference of B7H3 and B7H4 and PD-L1 across different patient tumors?
We looked at about 33 epithelial ovarian cancers, and it was very reproducible in our batch of
Okay, so
samples.
no doubt about that. Okay, very
Yes.
reproducible. Okay, good.
Yes.
So another question is actually related to the expression of of PD-1. You basically showed initially and in the beginning in one of your first slides the response to PD-1 blockade in different type of cancers, and then in terms of the response of to antibodies to PD-1 blockade, and you've considered the differential expression depending on the cancer type. But how about within each type of cancer? Would we see a difference also inter patients? I mean, when we consider different patients, I mean, in other words, should we consider a type of, each patient will respond differently depending on the type of immune checkpoints that he would express, and we will go to sort of personalized, medicine sort of. Could you tell
Yes.
us more about that, please?
Yes. So of course, so was what we thought we were going to see, that there would be so much heterogeneity between patients that we wouldn't be able to make a conclusion about, say, regulatory T cells from ovarian cancer. What was very striking to me, and I almost wanted to publish 10 individual patients and what their profiles look like if we stain for PD-1 and FoxP3. So one of the first slides I showed, it was like every patient, it just looks like that. And it was for us very, very remarkable in terms of how common a certain marker phenotype was in a certain cancer. So you would think the heterogeneity should overshadow everything, and it really didn't. At least I can say very, again, confidently for looking at, for example, PD-1 and FoxP3, or PD-1 and ICOS or something like that. So when we were characterizing the Tregs from ovarian and melanoma, they were very, very distinct.
Okay. Thank
So
you, Pamela.
very surprising to me, and I don't know. Yeah, viably frozen samples and thawed after whatever amount of time, but they still maintain that very clear phenotype.
Okay, another question. You tell me when you're tired, Pamela, because
well, if there's questions, we'll just keep.
Yeah. Two or three more questions and then we'll stop. Okay,
Yes.
2 or 3 more and then we will stop. So another one is how to explain that PD-1 high Tregs are highly suppressive, but that the blockade of PD-1 increased Treg frequency and lead to hyperprogression in patients.
Yes, so I think that's a hypothesis. It's been shown by another group, and we also were trying to test whether PD-1 had any impact on Treg function. So we were taking our ovarian Tregs that are expressing PD-1 and putting anti-PD-1 on them and looking for something. We weren't unable to find that, but that one paper, and I've seen other papers since, but the one I was highlighting in PNAS supported the idea by looking at the biopsies that there was proliferation of the Tregs. So generally speaking, there's quite a lot of proliferation of Tregs anyways, but in their case, they really quantitated the number in the tumor. So I think that's correlative work that says that that might happen. I believe, if I think back, I think they also showed, and I have seen, in other papers, that actually if you add anti-PD-1 to Tregs, they will proliferate more. And the other comment I would like to make, though, is that many of us are so used to the literature saying PD-1 somehow changes exhaustion and reinvigorates T cells. But actually, if you go back and you read the patent literature, okay, how did they actually come up with these antibodies? How did they select these antibodies? The patent literature says that, or shows, they were screened on inducing proliferation of T cells. So they were actually taking T cells, adding SEB, so staphylococcal enterotoxin B it's something that promotes proliferation of some cells, and then they were adding their antibody on top of that. And they were selecting, all these antibodies were selected for increased proliferation of T cells. So in my mind, it doesn't really surprise me that we could see a phenotype that's associated with increased proliferation of T cells. And then when you look at some of the genomic sequencing data about the TCRs and the repertoire shifts once patients have been treated with PD-1 blockade, again, that's consistent with the idea that certain clones are expanding upon PD-1 blockade. So I think for me, it's all consistent with the idea that you can get expansion, whether it be Tregs or regular cells. For me, that's one of the things that this antibody does.
Okay. Another question is related to these slow versus fast growing cultures. So the question is, how were they established, actually, these cultures?
Okay, so they were established exactly as the Rosenberg protocol is for expanding TILs. So we originally did these expansions because we were asking the question whether or not we could think about doing adoptive cell therapy using TILs from ovarian cancer and different kinds of cancers. And that's what got us to even looking at these expansion cultures and whether they were fast or slow. So the details would be check any Rosenberg protocol, but that's how it's done. But basically, smash up a tumor, small pieces, or digest it enzymatically, put them in wells with IL-2, and that's what happens.
Yeah, and a last related question actually to this slow growing, cultures. Do you see a lot of eosinophils in the slow growing breast cancer?
We haven't looked carefully enough. I think that's an interesting question, and whether or not they would actually persist in the culture over time, I don't know that the culture conditions would allow them to grow, but I think early on, we should look to see if there's a correlation between did it come from a tumor that was enriched in eosinophils. So yeah, that's a great question.
Yeah, okay. Thank you very much, Pamela. I think we will stop here.
Okay.
And I thank you very much for answering all these questions. And I thank everybody for being with us today. Thank you, Pamela, again.
Okay, thank you too.
Bye-bye,
And thank you
and
for
see you
everyone
soon.
joining
See you.
today.
Bye-bye. Bye-bye.
Bye-bye.
Bye-bye. Bye-bye.